Hello, I am Mahmud, a Master of Research student. Today I am going to talk about my project proposal, and it is on soil organic carbon dynamics in Cumberland Plains Supersite. My supervisor is Professor Alice Pendle. In the first part, I will talk about why soil carbon is important, and second part I will discuss about my project related methods and impacts. Let's start. So, what is soil? Soil is a dynamic natural body on the surface of the earth in which plants grow and is composed of minerals and organic materials. The soil consists of four major components, and these components cannot be separated easily because they are very intimately mixed with each other. As you can see in the pie chart, soil organic matter is only 5%, but it plays a very critical role for the agriculture, ecosystem and environment. My project will look at this organic matter, especially organic carbon dynamics. Now, soil organic matter, it comes from living organisms. Soil organic carbon is a part of soil organic matter. This organic carbon and its associated compounds exhibits a range of physical, chemical and biological property of soil that ultimately supports and backbone of all living things on the planet. Now discuss about my project. My project is based on the soil of the Cumberland Plains Supersite. It is a site of Terrestrial Ecosystem Research Network, commonly known as TERN. It is situated next to the Western Sydney University Hawkesbury campus. In regard to my project, the use of land of this supersite is identified into three categories. These are grassland. In the map it indicated with the black lines. Another division is woodland and marsh. On my project, at the moment I have four research objectives. The first objective is estimate soil organic carbon stock and other nutrients of the site on land use types. And I will be testing a hypothesis: is there any significant change in soil organic carbon stock with the land use types? Second objective is determine what drives soil organic carbon, considering a variety of soil and environmental variables like nutrient availability, texture. Third objective is development of a near-infrared library for rapid and cost-effective prediction of soil organic carbon and associated parameters like clay contents. And the fourth objective is to find the decomposition rate via incubation experiment to test is there any difference in rate because of conversion of land. Now talk about the soil sampling. A set of samples were already collected in early 2015. Another set of random sampling is proposed from 30 locations from the site and from two depths is 0 to 10 centimeter and 20 to 30 centimeter. In the map, the yellow dots are the point from where soil samples were already taken, and the planned soil samples will be from the location where it was not taken. Now the methods. I am going through these methods very briefly. Fresh soils will be analyzed for available nitrogen, ammonium plus nitrate on potassium chloride extracts followed by colorimetric analysis. Phosphorus and potassium will be measured according to the Colwell methods. And soil organic carbon and nitrogen stock will be measured on ground samples by combustion on an elemental analyzer. For the third objective of this study, to get the reading from the NIR machine, first sample needs to be to make into fine powder using a mill. Each sample will be placed in the cups and milled for 2 minutes at a frequency of 30 Hz per minute. These samples then be analyzed using the NIR with a FOSS XGS rapid content analyzer over a range of wavelengths from 700 to 2500 nanometer. This spectra will be analyzed by free software named R using required packages. The intended incubation method is to determine Turnover dynamics of carbon pools. The soil sample will be sifted to 4 mm to remove the gravel, roots and above ground biomass. An amount of 20 gram of sieved sample will be placed into a cup and will be brought into 60% fill capacity using deionized water. The cups will be placed into a 500 milliliter canning jar in a dark place at 23 degrees Celsius temperature. A 20 ml of head space specimen will be collected using a syringe at several intervals throughout the incubation period, and carbon dioxide concentration will be analyzed using a gas chromatographer. The data gathered will be fitted into a two-pool decay model equation to get the carbon pools and decomposition rates. Now, impact and scope of my proposal. The data from the study may provide other research with an additional or background information in that area. The government agency could get the data to make soil map around the area. This experiment result interpretation may be commercialized to determine commercial samples. The soil organic carbon management can restore the ecosystem and provide enormous aesthetic benefit to the society. Another important feature of the study is that it will measure soil organic carbon through two different methods. This may validate the data by comparing the results. Although the amount of soil organic carbon present in the soil suggests a signal of soil health, but the limitation is that it is very difficult to suggest an optimum level of soil organic carbon, as the soil is a very complex substance and the variations of climate. Moreover, the potential of carbon sequestration and management can help to mitigate climate change effects. In conclusion, I would say soil organic carbon is the backbone of the human civilization, so we should study it more for the benefit of the society. Thank you very much for watching. Goodbye.